Illuminating the microscopic world, fluorescent antibody labeling. We live in a universe teeming with life, from the smallest microbes to the largest whales, yet much of this life remains hidden from our view, obscured by the limitations of our own senses. And to truly understand the complexity of life, we need tools that can unveil the secrets of the microscopic world. This is where fluorescent antibody labeling, or FIA, comes in. FIA is a revolutionary technique that empowers scientists to visualize and study biological processes in unprecedented detail. It is a cornerstone of modern biotechnology, with applications spanning medicine, agriculture, and environmental science. Defining FIA, a powerful tool for biotechnology. FIA is based on a simple yet elegant principle, using antibodies as molecular flashlights to illuminate specific targets within cells and tissues. Antibodies are proteins produced by our immune system to identify and neutralize foreign invaders, such as bacteria and viruses. Each antibody binds with remarkable specificity to a particular target molecule, known as an antigen. In FIA, scientists exploit this specificity by attaching fluorescent dyes, or fluorochromes, to antibodies. These fluorescently labeled antibodies then act as guided missiles, homing in on their target antigens and making them glow under a specialized microscope. Antibodies as molecular beacons: structure and labeling. To understand how FIA works, it's helpful to visualize the structure of an antibody. Imagine a Y-shaped molecule with two arms and a stem. The tips of the arms are the antigen binding sites, each tailored to latch onto a specific antigen. The stem, known as the FC region, is where the fluorescent dye is typically attached. This labeling process is carefully designed to avoid interfering with the antibody's ability to bind to its target. The fluorescent dyes used in FIA come in a rainbow of colors, each emitting light of a specific wavelength when excited by a laser. This allows researchers to simultaneously track multiple targets within a single sample, painting intricate pictures of cellular architecture and function. The FIA process, from sample to stunning visuals. The process of performing FIA is akin to developing a photograph. It requires careful preparation, precise timing, and a bit of artistry. The first step is to prepare the sample, which could be anything from cells grown in a petri dish to thin slices of tissue. The sample is typically treated with special chemicals to preserve its structure and make it permeable to the antibodies. Next comes the staining step, where the fluorescently labeled antibodies are added to the sample and allowed to bind to their targets. Finally, the sample is washed to remove any unbound antibodies, and then placed under a fluorescence microscope. The microscope illuminates the sample with a specific wavelength of light that excites the fluorescent dyes, causing them to emit light of a longer wavelength. This emitted light is then captured by a detector, generating stunning images of the labeled targets against a dark background. These images can reveal the precise location of proteins within a cell, track the movement of cells within a living organism, or even diagnose diseases based on the presence or absence of specific markers. In conclusion, fluorescent antibody labeling is a transformative technique that has revolutionized the field of biotechnology. By illuminating the microscopic world, it allows scientists to explore the intricate details of cellular processes, leading to groundbreaking discoveries in medicine, agriculture, and environmental science. As we continue to advance this technology, the potential for new insights and innovations is limitless. Imagine a future where we can diagnose diseases with pinpoint accuracy, develop crops that are more resilient to climate change, and monitor environmental pollutants with unprecedented precision. The applications of FIA are boundless, and the journey of discovery is far from over. Thank you for joining us on this journey into the fascinating world of FIA. Stay curious, keep exploring, and together, let's unveil the mysteries of the microscopic universe.